Thank you so much for this introduction. Thank you all for still being here. I would like to continue the interesting topic, an interesting dive into plant biology that we had at the moment of plant photobiology. I would add additional complexity to this picture because plants do not work alone. They have some soil allies. So the topic would bring some of the examples, some of the science that we already did quite extensively for the agriculture sector, and use that example to think about some good recommendation practices for the turfgrass sector as well. But at the beginning, I would like to take you for a bit of the journey. A journey about 200 million kilometers from the Earth's surface to that small red dot on the night sky, to Mars. Why would I like to do that? Because if we consider if ever our society would be able to do a space farming project on Mars, we could be sure of one thing, that this environment that consists of 95% of carbon dioxide in the atmosphere with just a little traces of air, argon, and other gases, no organic matter, high UV light exposure, all of that doesn't favor any form of life. But there was the NASA trials that use specific extremophytes, so bacteria that could tolerate extremely harsh conditions to see if they could favor some of the processes controlled on their behalf. And they did it, of course, not on Mars, but on at least very harsh Earth conditions based on hard volcanic ashes, that without any nutrient surplus, with high UV light and high temperatures, they are able to survive, sustain the life, and bring that foundation for the life. But let's get back to the Earth with still the carbon dioxide content of 0.04%. We still have in agriculture quite a lot of the problems. So we still try to use some of biological complexities to try to improve the productivity and improve the sustainability of the produce. But taking into consideration the agriculture as the model of business, we have quite a lot of uncertainties. The same like here, some of the examples when harsh environmental conditions in sub-arid areas for the turfgrass sector as well affects pretty badly the performance on the quality grasses. But what if we try for the agriculture, we try to model all of the fluxes? There was a research in Ghent University that tried to model all kinds of fluxes for the agriculture, on the inputs, outputs, try to model to understand the complexities. And it only resembled us the complexity of the Tokyo subway station. So trying to put it as a kind of the big picture doesn't make sense. So maybe we try to have a different options. So going back to the picture, like to try to understand what the complexities of plant biology are, what struggles could we have, we could try to reverse that perspective. So start with the soil-centric approach. That's the way. That's the way of thinking that is quite in line with my background. I did quite extensively work in soil biology as plant nutritionist and also for human toxicology doctor. Starting from healthy soil, that's the core foundation of productivity of plants. Why so? Because in soil we have that amazing biodiversity of the microbes, but also other biota living there. We have a long evolutionary pathway that the soils in natural areas go through to form a sustainable place for the root to sustain all the conditions, provide the nutrients, provide the access to the water, and provide the space for the plant to be into the ground. We have that bacteria that are in concenter of the nutrient mobilization processes. That would be one of the topics that we'll cover and try to show you in the light of what we could do with the biologicals based on some of the experiences for the agriculture sector, and of course the microbials are also in the core center of the relation, of the adaptation of plants of any kind to the climatic challenges. So they are needed to build the soil, to sustain the soil in the right conditions. And of course, the numbers are the basic one that I use. The healthy soil coming from the mature ecosystem is a rich library of life. We could count between 100 to 800 million bacteria cells classified into roughly around 10,000 species. We have a few kilometers of fungal hyphae. That's for boreal forest. For agricultural setting, could be half, could be 1/3 of that. We have also between 10 to 30,000 protozoa. We have 50,000 of algae cells. That's still within just 1 gram of the soil. We have between 20 to 30 nematodes. And believe me, not all of them are bad. Some of the nematodes are also quite crucial for sustaining the balance in the soil. And we have around 5 annelids, mostly airborne. And in that space, we have a very specific interface. Let's take an example of that Rubik cube, that on every axis, we have first the plant genotype, that is crucial, plant control by photosynthesis, the carbon fluxes inside needs its proper line to adapt to the conditions, but we have also a complexity of soil, the parameters related to its physico-chemical concentration, to the validity, the form how it shapes, and the third axis, the microbiome. Microbiome, that is one of that third diversities here, and around that we have a lot of uncertainties. So if we would like to fix that Rubik cube, trying to find the best recipe for the growth of crop, we need to face that process, like try to fix that Rubik cube, but doing it in the dark when we cannot recognize the color. So it's very hard to try to merge all that complexities into one picture. But we could try to at least find some guidelines, find based on the research that was done, that is available, and try to use that insight to find the way how we could use that biologicals, that biocentric approach to improve the productivity. But to do so, we need to very briefly focus on what really that complexities of the soil are. Soil is very specific substrate. It's different than artificial substrate of growth. It is consist of very specific aggregates of different size. So we have macro aggregates. So they are the big one in healthy soil. They could have more than centimeter in size. They are quite crucial for the water filtration because they are physical statues. So they filter the water, release the contamination. They are also the place of living of fauna and the roots of plant. Between them, we have a water-stable aggregates of around 1 millimeter, so we'll need some magnification to see them, and they are crucial for both. Again, they act as a sieve to filter the water. They are also responsible for the physical stability of the soil, and they start to be a habitat, place of living of microbials, so microorganisms. Then we have microaggregates and elementary soil particles, critically important for the water storage, for chemical buffering, and also for the carbon storage in the soil. So we need to take into consideration that physical stability is closely related to the soil life that is in the soil. And some of the processes are highly manageable to try to find a shortcut by using the synthetic inputs. And there is interesting fact that if I will tell you that plant roots and microorganisms in the soil communicate with each other. They do so. They, of course, do not do it verbally, but they do it with the chemical signaling. What we have seen, like in the previous lecture, we have that night shift during plant growth when it uses the sugar. So one of the uses is to release exudates. Exudates, that are substances that are released from the epidermis to the rhizosphere, and there are the chemical signals that plant send to the surrounding, and the core recipients are bacteria and fungi that live in the soil. And bacteria and fungi that live in the soil, both pathogenic and beneficial one, are the core responders and controllers of the processes. So they send back also the signals, being fed with that chemical, mostly sugar, but also some other secondary metabolites. They respond, and they could help plant to sustain its growth in the soil surface. So let's see where that whole situation happened. As a unique video done in Netherlands of the growth of roots with visible root hair evolution. Root hairs are produced by just single cell panicula that grow from the epidermis, and there are the place when plant absorb the water. Not with the core surface of the root. They mostly form the interface for nutrients uptake, for water availability, and also interface of that interaction with the microbes. A single plant, single grass plant or single cereal could have more than 30 kilometers of root hairs in length in mature root. So that's unique micro world for a lot of the interaction that happen in the world. And as I tell you previously, we have quite a large diversity of the macro, meso, and microfauna that live there. And every part of that biological diagram has its own role. We have the microfauna consisting of bacteria, fungi, nematodes, protozoa, and rotifers that are responsible for the decomposition of the biomatter, mineralization, carbon storage, and disease control. I will have a little focus on that process a little bit later on. Then we have a mesofauna responsible for organic matter fragmentation, micropore formation, aggregation, stabilization of the aggregates, especially the water-stable aggregates and microaggregates, and also for predation. There are the next level of the trophic chains needed to release the carbon again into the system. And then we have a macrofauna responsible for bioturbation, so mixing the biomatter, also the formation of the macropores, responsible for the improvement of the soil structure, and also redistribution of organic matter in the system. And in that picture, we have different spheres. No matter if we are talking about the farming crops of the turfgrass, that's all really the same. We have the rhizosphere, that's the part of soil that is under the direct influence of that exudates, so all the substances released by the root. We have a detritosphere, that's the part when we have a dead biomatter, but below the soil surface, not on the front, and we have the bulk soil. So the richer life is here. Still, we have a lot of life in the middle, and the bulk soil is the reservoir of the other parts. And between that all spheres, we have mycorrhizal fungi, different kind, and that mycorrhizal fungi are like highways for transferring water, for transferring nutrients, and it is also a highway for the bacteria. You could see here that when it zooms, there is a flow of bacteria on the biofilm or hyphae. That's important part, even for the plants that doesn't form a symbiosis with fungi. They need to have its presence for transmitting, for giving the space for the bacteria to follow that pathway of the microfilms. And saying that, we do not have a fully homogeneous soil surface. When we take different geographies and different sampling paths, we have a different microbiomes, so we cannot put it into one bag. So, of course, soils containing the mineral matter, namely sand, slate, and clay. We have organic content, we have microbes, air and water, and we have a recipe for the soil. But it differs around every root systems, and it differs in different climates, and it differs in different geographies. And you could only imagine the whole complexities for biologicals if for every sampling spot we have a wide array of interaction. That research has been done for selected sampling sites, analysis of the metagenomics of the bacteria that live in that spot. And you could see with the red lines, there are the negative interactions. With the green lines, we have a positive interaction. And both of them are crucial, because without negative interaction, we would have no negative feedback loop to control some of the microbes. But what's the reason for showing this is there are not easy answers. So for agriculture sector, it has been extensive amount of research, mostly concerned into very stiff and very artificial testing background that tried to use that biocentric approach to improve the quality of soil. And there are specific genera, specific species of bacteria that control a lot of the processes. I've given here the examples of most prominent one used in a lot of market biological products that we have, for example, for nitrogen fixation, specific strains of and species of bacteria, both symbiotic and free-living bacteria, that could improve the fixation of nitrogen from air, and it could help to reduce up to 30–40% of synthetic urea input. But also we have bacteria that helped in solubilization of phosphorus and potassium, bacteria and fungi that could, by using its physicochemistry, use that resources from organic and non-organic sources to put them into a new equation and allow its availability in the soil. Then we have specific bacteria and fungi involved into phytohormone synthesis, so they can directly interact with the plants and favor some of the growth processes. Next, we have also specific bacteria that are involved into the pest control and pathogen control, involving nematodes, pathogenic bacteria, and insects. Very hard to register in Europe, but there are some formulas that uses natural-based mechanism of that living organism to release some of the biotic stresses. And last but not least, we have also specific genera of bacteria and fungi that could effectively help in the degradation of organic, but also non-organic pollutants to store them properly. For example, something like heavy metals that by the use of siderophores could be precipitated from the soil to get rid of some of the contamination. And that the bacteria, as nitrogen was taken into the first example, are in the core center of the biological processes of nitrogen metabolism in the soil. We have bacteria that's mainly all about like changing the oxidation status of nitrogen. Not all forms of nitrogen are available by the crop, so starting from that nitrogen fixation, so we are adding nitrogen to that equation. Then we have processes of nitrification and denitrification, so going along the cycles, and the assimilation of nitrogen is only available by taking nitrates to change and transform them to the organic forms and put them into the biomass in the forms of amino acids. Also, bacteria are the core players when it comes to the mineralization, bacteria and degrading fungi, to decompose the biomatter and again put nitrogen back into the system. And that's an interesting example because, of course, nitrogen is the widespread. You could use an extensive amount of course, like for the agriculture sector, that's a stress factor. Here for the turf grass maintenance, probably not the core price-making factor. But if we overuse the urea form of nitrogen, we are changing the situation in the soil fiercely. First of all, the soil that is strongly dependent on the external input of urea wouldn't favor a proper development of the root systems, because plant demands the regular application of nitrogen, so there is no force that would reshape root architecture in the way to favor some secondary growth adjustment. We have a strong tube that transfers nitrogen into the soil, and we have a tank that is broken because around 60 to 70% of urea nitrogen is lost through the soil. That, of course, for the agriculture sector poses significant sustainability issues. But that's another way how we could improve that nitrogen balance. We could invest in that soil biology. So still, we would be dependent on nitrogen, on complex fertilizer, other forms, biofertilizer, whatever, but we would favor that biolife. So by that, if we invest in soil biology, we could increase the production of root exudate by crops. They form that close relations with symbiotic bacteria and fungi. Symbiotic bacteria and fungi allow root system to get inside of some nitrogen resources in the packages inside the soil. We are also favoring that next trophic levels by the predation by rotifers, other mesofauna, and we are getting additional pipes that bring the nitrogen into the system. So we will still be having the losses because we cannot store nitrogen into the soil due to the precipitation or due to the use of watering system. They will be also getting rid of part of the nitrogen, but we have additional pathways that recirculate the nitrogen into that system. And as I told you before, also bacteria and fungi are the core players when it comes to plant residue decomposition. Basically, that's the biggest resource we have on the farming field, or we have the cutted grasses by using enzymes or by using specific microbial consortia. We could get that lignocellulose breaking down, and that's one of the biggest resources of nitrogen in the agriculture setups. So microbes or enzyme derives of microbes consist of cellulose, xylanases, proteases, so whole arsenals of enzymes that use that biomatter to change that transfer into products. Products consisting of nitrates, glucose, pentoses, so basically sugars for the crop, amino acids, alcohols, and nutrients. So by wisely selecting the microbial cultures in the way of the inoculants or in the way of the agronomic practices we use, we could make the use of that resources that are abundant on the agricultural setup. And there is also one mechanism that crops or other plants could make advantage of. That mechanism is called cry for help. And basically, plants are the one to cry for that help. So look for allies to help them to sustain some specific stresses. It could be abiotic stresses, it could be biotic stresses. So plant change its exudate profile. So change chemistry of produced compounds to attract specific microbial helpers. And it has been tested, but for example, the release of malic acid, fumaric acid of tryptophan can trigger and allow the recruitment of specific beneficial bacteria like Bacillus subtilis or Bacillus amylolicefaciens, the one living already in the soil, to fight some specific pathogens as Pseudomonas syringae, the leaf pathogen, or Fusarium, that's the root pathogen, by the bacteria that are already in the soil. Similarly, the release of strigalactone as that alarming bell of the crop is the way to recruit arbuscular mycorrhiza to protect against the water stress or low temperature stress. The other effectors are when the bacteria favor the metabolism of phytohormones and that indirectly help to induce the systemic resistance. So quite a lot of the functions, but if we let the biology of the soil work, that we could allow to reduce some of the inputs and make the savings mostly for the resilience of the plants. So that's main goals for the agriculture. So on the left-hand side, we could say that if we use the biologicals for the increasing of root emissions or non-volatile secondary metabolites, or increase the volume of volatile organic compounds, we have that signaling compounds that can be used as exchange of the pesticides. So we are reducing the need for plant control agents. On the other way, if we invest into the use and increase exudation profiles of organic acids by adding biologicals to the system or adapting agricultural practices that would favor the soil health, and we also increase that crosslinks, positive crosslinks between rhizobia and plant roots, we could also improve the nutrient availability on the mechanism that I showed and reduce the dependence of fertilizer. That still they would be needed. They wouldn't be such huge stress factor for the crop. So trying to find the right balance between the use of that soil applied fertilizer. And when I talk about the use of the microbes, they are not plug and play solutions. So the old approach was to try to indirectly modify the soil to favor some specific microbes. So by farming practices, by keeping the soil covered, by allowing deeper or shallower tillage, we are affecting the microbes that live there. The same different kind of soil amendments, so using the biomass, using watering schemes, we are also indirectly modifying the microbiome of the soil. We could use inoculates, and the old approach was like in mathematics, that we are isolating one microbe, putting it into hostile conditions. They are hostile because they are different than the lab one, and trying to see if that would work. In many cases, that wouldn't. Also, the use of fumigants for the pathogenic fungi completely changed the balance of the microbiome of the soil. More advanced approaches for this method to use the biologicals, as they use to customize the inoculants. So they use specific probiotics with prebiotics, so that secondary metabolites of the crop, to use more customized solution for the crop. And the novel approach is to focus on consortia of bacteria and fungi that will be functional. So focus on the conditions that the crop face, and focus on specific physical chemistry of the soil to provide a customized probiotics and prebiotics to fix the issues of certain situation that we have with the plant. And also we have a wide range of available biostimulants for the turfgrass maintenance sectors that consist. Here on the one chart, we have the beneficial microbes. We have also some prebiotics as humic and fulvic acid, seaweed extract, and also some beneficial elements. And I have did this summary based on the meta-analytics that use that for different species of grasses. We could improve root phenotypes. We could improve the availability of the nutrients. We could improve the dry matter content by trying to find the right spot between using synthetic consortia, feeding them properly, and adjusting agricultural practices to serve that purpose. And by the use, as the take-home message, what I would like to tell you is not an easy solution. That's not, microbes are not a universal key that fits to every door. We need to carefully focus on the research that has been done for similar conditions, for similar plant genotypes, and also focus on the process that's the problematic one that we need to find a solution for. So there are quite a lot of the components, raw materials used for the biostimulants, all the microbial-based one, or prebiotics or postbiotics that could help in the access to limited nutrients for central, or the loss of nutrients, precipitation of them, limited water as resources, abiotic stress exposure as high or cold temperature exposure, or salinity levels, or specific biotic stresses. And there are the mechanisms that we could favor and specific components. So as take-home message, I would say that still a lot of the investigation for the turfgrass sector to see if the biologicals would be a remedy for some of dependencies of chemical inputs. But at least it's worth a trial, because I told you the microbes are the one that could help in the long term in having the resilience of the production. There was a Chinese research published this year showing that due to the climatic change, due to extreme in the climate, we are in the risk of losing 1/3 of productivity of rice in the hotter climates, and there is assumption by investing in soil health. So generally, by having a higher abundance of the microbes and having the higher activity profiles in the soil, we could at least control around 20% of that loss. So reverse that challenge. So we are investing, by investing in soil health, we are building that long-term resilience, because we cannot know, we know that the extremes would be there. It wouldn't be an easy way, but I would like to leave with these conclusions that the methods to alter soil health are not a new one. That's not a state-of-the-art biotechnology made in the labs. It is technology that was already used in the ancient Romans. There was a practice of dipping the seed material of grasses and grains for the cereals in the seaweed extracts. They didn't know why they did that. It works for the very low water resources seasons, and that was a prebiotic for the microbes. So the science brings us the answers for some of the activities. We just need to employ that science to serve the purpose. Thank you so much for your attention, and I would be very happy to answer any additional question. Yes? That would be a definitely limiting factor, because with the biomass lower than about 30% of carbon allocation in the soil, it is very hard to allow a single inocula survive. But for this reason, mostly does it work? Mostly the use of microbial and fungal inocula could work, because fungal has a very higher capacity of degrading and producing that biomass that allow then bacteria to survive in the soil. But that's a critical matter.
Is there any simple way for us to measure microbial activity in our soils?
That's a good question. I'm struggling with that pretty fiercely in different climates. I have done all kinds of methods of analytics, from metagenomics to enzymatic profiles to very simple microscope-based approaches. For me right now, the most promising one are the use of enzymatic profiles. So we could use basically the plates with different carbon source and score what's the functional diversity of the microbes. That's more like plug-and-play solution for testing. I did a project. It's now running in Lithuania, in Kaunas, in the public space that we do the test on reducing the synthetic inputs, doing microbial testing on many different crops, 8 different crops for Central European climate zone, and trying employing quite advanced analytics to compare metagenomics, compare that enzymatic profiles and abundance of the bacteria, to see where would be that sweet spot that we could reduce some of the fertilizer, add microbes, save on everything, and still have a very good productivity. Okay, so thank you very much.